Okay, now I'm going to do what I actually came for. So it's really my great pleasure to introduce my friend and colleague, Dan Tagle. Dan is currently the, I'd say, Associate Director of Special Initiatives, but he's also acting as a Deputy Director at this point of the National Center for Advancing Translational Science, or NCATS, which is the institute that provides the CTSA awards across the country. And Dan and I have worked for the last 5 years as part of being a council member at NCATS. We've had a lot of different opportunities to work with each other over the last 5 years. And of course, I see him 3 to 4 times a year whenever I go to NIH. He is really the embodiment of what you would call translational science. He got his PhD from Wayne State, and then did his postdoctoral work with Francis Collins at University of Michigan, and then went with Francis to NIH. And there he's had senior leadership role at the Genome Institute, and then was a senior leadership role at the National Institute of Neurological Disorders, or NINDS, until NCATS was formed, when he became a special director of special initiatives with the director's office. He has served in many, many roles within NCATS. He is the technology guy that NCATS and NIH goes to. And he's developed a number of initiatives specifically looking at organoids, tissue on chips, and variety of cutting-edge technologies that I think are going to revolutionize the biological discovery as well as drug development programs. So it's my great pleasure to invite Dan, who is is going to be talking about some of the tissue chip and organoids work that they've been doing. Please welcome Dan.
Well, good morning, everyone. And certainly a warm congratulations to Anantha. Very well deserved. He's being recognized for his contributions, certainly here in the state of Indiana. But as he had said, he works tirelessly as a member of the NCATS Advisory Council, where he's performing at the national level in terms of advising the NIH, which is a federal agency, in terms of achieving our missions. So I come here certainly thankful to Anantha and to Lori for the invitation into BioCrossroads. So in my capacity as the Associate Director of Special Initiatives, I do develop and implement and manage a number of exciting and innovative technologies and platforms that would influence the life sciences. But I come here to you this morning to talk about specifically the Organs on Chips program. Just to give you a little bit of background about NCATS, it was established and authorized to be formed by the United States Congress back in 2012. That's actually when Anantha came to start serving at Council. And essentially, the United States Congress acknowledged that there are certain challenges and difficulties in the drug development pipeline. And so creating an entity within NIH seems to be a crucial mission to advance human health, especially US public health. And so our mission at NCATS is to generate innovative methods and technologies for all diseases. And so the current challenges in drug development, I think this has been alluded to in previous talks last night, is that it essentially takes, on the average, anywhere from 10 to 15 years to bring a drug to market at the cost of $2.6 billion. Now, added to this cost are all the failures that happen in terms of drug development. And these failures are largely due to efficacy or safety signals that would be toxic in humans. And part of the failures is because the current paradigm that we use in drug development essentially uses monolayer 2D cell culture systems as well as in vivo animal models. And so the way that we are trying to approach this is more on a precision medicine approach, which is not the one-size-fits-all, and essentially to come up with better predictive tools for risk assessment. And so this is where representation with drug development pipeline, showing all the cell culture and animal models, primarily in the preclinical space, where most of the failures happen, as well as in the clinical area. And so all the work is being done to advance the technology or the drug candidate before it comes to human, the first in human use, essentially are not serving its purpose in leading us to better and safe medications. And so this is where the NCAT's tissue chips or microphysiological systems program comes in. Essentially, it's to develop an in vitro platform that uses human cells and tissues in combination with advances in stem cell biology, in bioengineering, microfluidics, toxicology, pharmacology, essentially to come up with a better predictive tool. And so using these bioengineered devices, we will represent or have been representing major human organ constructs shown here, about 10 major organ systems. They need to be physiologically relevant. That is, it mimics not only the structure, but as well as the physiological function of the major organ systems. And of course, has to represent the genetic diversity of the population, of the US population. It is intended to be engineered to be modular. That means it's kind of like a Lego blocks that you can plug and play, these systems. And I'll show what the implications of that would be. And then, of course, tissue viability. Most cell culture systems would be viable for 2 to 4 days. And so we require that these systems actually be viable for at least 4 weeks. We have systems that will be running and alive for 3 months. And so you can actually do chronic testing of drugs on these systems. And so what do they look like? So this is a representative tissue chip. This is actually one of the first ones developed. It is the lung on a chip system. It's about the size of a USB stick. I actually have one right here in my hand. Right now, it's made out of a very flexible biomaterial, PDMS. You can twist it. But essentially, you take the scaffold, which is essentially the extracellular matrix, be able to configure that such that it support many cell types. So this is not just a monolayer. This is actually representative of whatever cell, different types of cells that are in particular tissue, the structure in terms of porosity, the rigidity of the tissue has to be represented, spatial and temporal patterning, which means cytokine, hormone gradients, oxygen gradients has to be represented, perfusion, that means vasculature, and these three-dimensional tissue systems. The bioreactors have to also incorporate the biomechanical stress. So if you have a bone on a chip, it has to have the actual pressure, or if it's a lung, which I'll show later on, it has to have the actual mechanics of a breathing lung. Of course, innervation, immune response, functional readouts, which entails non-invasive real-time imaging technologies, as well as omic technologies, essentially built into the platform, and then be able to have microfluidics be part of this system. This is an NIH program led by NCATS. We started this program right at the inception of NCATS in 2012, and just primarily focused on, for the first 5 years, on safety and toxicity assessment. We had partners from pharma, primarily AstraZeneca and Pfizer, and GSK, who we formed memorandum of understanding, essentially to provide us with a training set of compounds, essentially to replicate what those human reactions have been to this training set of compounds. This training set of compounds are essentially compounds that have sailed through preclinical testing, have been tested in humans, and of course failed. And so we wanted to find out if the tissue on chips actually were able to predict a human response that the animal models and the 2D cell culture systems were not able to reproduce. We also formed partnerships with DARPA, which also contributed financially to the program, and their goal is primarily to develop the 10-organ bioengineered platform system. And of course, the FDA is a key stakeholder in this arena because we essentially would want to have regulatory agency buy-in in terms of qualifying these devices for drug development. Certainly very productive in terms of publications over the last 5 years, but we were not certainly satisfied with that. So I'll tell you a little bit more in terms of what we're doing. But here's sort of like an example, a graphical representation of what the tissue chip would do. So here's a human lung. And so what would it take to represent a human lung on a chip? And so essentially, you bring it down to its basic functional element, which is the alveoli, or the air sacs. And to the left of the air sac would be an actual chip, which is the one that I showed you, which is this one, where you have channels on each side with vacuums. And cyclic vacuuming actually stretches the membrane where lung epithelial cells on top are shown in green, and then the endothelial cells shown in red in the bottom, which would be the capillary or the vascular side would be formed and actually would be stretched. So the actual stretching enables the lung epithelial cells to actually form surfactants. If the same cells, if you culture them two-dimensionally, would not even produce a surfactant, would resemble essentially very flat cells and would not resemble lung epithelial cells. The other thing that's been developed would be a microvasculature in the chip. Shown here are 2-micron beads, fluorescent beads coursing through a microvasculature in the chip. And you can actually embed tumors or other organoids into the system. To the right of that is a gut on a chip, which was innervated and actually would undergo peristalsis. In terms of non-invasive readout, this would be a liver on a chip showing changes from red to green, which is a fluorescent indicator of apoptosis or cell death as a result of mitochondrial membrane permeability. Here are representative organs on chips. The one on the top right would be the lung on a chip. The middle would be the microvasculature on a chip, and the far right, or my right, would be the kidney on a chip. The bottom left would be the female reproductive system on a chip. So as you know, in drug development, it's usually the male cells or the male species that's always been tested. So here's an opportunity for us to actually represent female gender in terms of drug testing. So in that particular chip, we have representations, the fallopian tube, the uterus, ovary, the vagina, and of course coupled with the liver to show how female hormones can actually influence drug metabolism and how it influences the liver. And then on the far right would be the liver, skin, bone marrow, and muscle, cardiac muscle. So again, just showing you examples of the diversity of different things that can be accomplished with this system. In terms of linking different tissue chips, we have certainly multiple organ chips on chips, and they can be linked and coupled together essentially to form the human body on a chip system, which can then be seeded with individual cells derived from individuals through IPS, induced pluripotent stem cells from individuals. So you can actually form a U on a chip system. This is President Obama when he actually authorized the Precision Medicine Initiative that Ananta is part of. And so we, at that time, in his last year in office was when the precision medicine funding got to be, and this is one of the things envisioned that the precision medicine will lead to, would be a U on a chip system where every individual in that cohort can actually be represented on a chip. Probably one thing I would emphasize in my presentation in terms of the take-home message are the partnerships, how critical that is in terms of making this innovative technology essentially take off and get to where it is at its current state due to the numerous partnerships we have. So we have partnerships with various government agencies, pharma, other institutes and centers at the NIH, nonprofit organizations, with space implementation partners through NASA, testing centers, certainly academic institutions, as well as startups that have spun out of these academic institutions to commercialize this technology. So I'll just briefly mention in terms of an overview what these partnerships have been. So as I mentioned, FDA was a key partner right from the beginning of the program. So a key sign in terms of FDA and pharma interest is, are they actually going to start using this technology? So just a year ago or so, FDA actually signed collaborative agreements with a couple of the startups, Emulate and CN Bio, to actually establish their own organs-on-chip facility right at the FDA. Likewise, AstraZeneca formed a partnership with Emulate to actually, for Emulate to set up organs-on-chip facility in their Swedish drug development facility. And then FDA also is looking now to have an input in terms of what it takes to get this technology incorporated into the drug development pipeline through the predictive toxicology roadmap. And so that's a public hearing that's still open to public input up until 2 days from now. An important partner will be is the IQ consortium. It's made up of 20 pharmaceutical companies right now. And essentially, this IQ Microphysiological Systems affiliate serves as an advisory body to NCATS, to NIH, in terms of what drugs to test the system, to validate them, and essentially to gain confidence into what it takes for the system to be adopted into the pharma drug development pipeline. So out of this work with FDA and the IQ consortium, we have formed what we call a tissue chip validation framework, where we're not satisfied with just the tissue chip developers putting out those publications. And so while they were very productive and in terms of what we call physiological validation, that is demonstrating organ function and structure, and then also showing that the training set of compounds show physiological response. We wanted to bring it a step up in terms of generating analytical validation, and this is done through testing centers, independent testing centers that would take the data, the platforms, the cells from the developers, and these are sort of like naive laboratories who would take this technology for the first time and be able to give us feedback in terms of how easy are the technology to be incorporated into their lab, how reproducible, and how they can replicate the findings of the developers, and essentially come out with best practice and standard operating procedures and what the best cell sources are so that this can be adopted quickly by the community. And then the last stage of validation would what we call the industrial adoption of this technology, essentially when pharma starts using them for their proprietary set of compounds. And we envision this to be more of a CRO-type environment. And this is where the adoption and commercialization happens. Falls in line with our NCATS 3Ds, which is to develop the technology, demonstrate its utility, and of course disseminate the technology. And as I've said, it was done with partnerships through FDA and IQ consortium. So these are the testing center goals. We have 2 testing centers, one at MIT and one at Texas A&M. Those are the independent testing centers. And then we created also a database to house anything that's chip-based technology. And then below are the organ systems that have actually been tested. Paper actually just came out this week in Nature scientific report on the first testing on the kidney on a chip, and essentially telling us what the lessons learned are in terms of this technology. Where we're going next with this technology is to shift from toxicity and safety testing to now disease modeling and efficacy testing. And so, of course, that always starts with the disease. And so whether it be monogenic, polygenic, Mendelian or multifactorial, single organ defect or multi-organ physiology, we take IPS or primary tissues, put that on the tissues on chips, where we get in vitro readouts that can then, of course, be correlated with in vivo clinical findings, be able to do in vitro and in vivo extrapolation from this data, and then essentially use that to inform the next generation of chips. So in this regard, we started working with rare disease patient community, which is an important patient engagement is always a mantra that NCATS and Anta would say that patient engagement is central to our activities at NCATS. So we work with patient advocacy groups to develop models for a number of rare diseases. So there's essentially rare diseases where you could not even find any animal model for them. And that also led to a full-blown program, which is now being supported for the next 5 years to develop various models for a number of diseases. That includes like psychiatric disorders such as Proteus syndrome and DiGeorge syndrome, ALS, Parkinson's disease, cardiomyopathies, kidney disorders, musculoskeletal defects, polycystic ovarian syndrome, again being done in a number of academic institutions all over the United States. One exciting thing is our partnership with NASA, and because we can learn a lot about the biology in terms of very accelerated ways because of microgravity. And so when astronauts go up in space, within the first 3 weeks that they're in microgravity, they actually experience upper body fluid shifts, ear, neurovestibular disturbances, sleep disturbances, bone demineralization. About 6 months or so, they start having bone resorption, muscle atrophy, cardiovascular deconditioning, GI disturbances, hematological changes, and then they have declining immunity, renal stone formation, all symptoms of premature aging. But the interesting finding is that when the astronauts return to Earth, they revert back to normal physiology within a few weeks. And so here's an opportunity for us then to learn about human biology in terms of accelerated aging under microgravity conditions. So we have projects that would look at immune senescence, sarcopenia, osteoporosis, proteinuria, kidney stone formation, conditions that would take months, if not years, to model here on Earth. But under microgravity conditions, we can actually mimic these conditions within weeks under microgravity. So here's a number of academic institutions and biotech companies that are funded to do this work. And the exciting thing also is that it's an opportunity for us to advance the technology. So I showed you how small the chips are, but what's usually a well-guarded secret is that it takes a very sophisticated contraption and control systems to support the chips in terms of incubators, pumps, valves, controlling oxygen levels. But here's an opportunity for us to partner with NASA engineers to a number of these companies through NASA in the cases, essentially to transform this technology that would support the chips into the size of a shoebox. And that's essentially accelerating the technology in a very rapid way by tapping into expertise. So when I talk to astronauts who have gone up to the International Space Station and actually will be running these experiments, they say, what sort of like take-home lessons can you give us? And said, we're pretty much 90% military, fighter pilots. And so anything that you can do to make it fighter pilot proof, if you can make it, run the system by just the push of a button. We don't need advanced degrees in engineering or medicine to run these experiments. And so anything that's turnkey would be helpful. And of course, we have to reduce the footprint. So this is not just speculation. So this is actually a project that started last year, about 18 months ago. So here's an example of a project on the kidney on a chip that's looking at proteinuria and kidney stone formation. So the chip itself is about the size of a credit card, where you have 3 kidney proximal tubules represented in that chip. The project will involve 72 samples to be taken up, so about 24 chips. The bottom line is that these are the requirements to run those samples here on Earth. So just the dramatic thing is, 216 feet of tubing, 4 incubators, about 48 cubic feet or so, and then just reducing that into a small footprint. So in orbit, that is to take it up to International Space Station, essentially we have to reduce it to the size of a shoebox, as I mentioned, about 2 cubic feet or so, which is SABLE, which stands for Space Applied Biology Laboratory on Orbit. And so just to give you a dramatization of what that looks like, the 48 cubic feet would be a support system here on Earth. In order for it to be launched in SpaceX, the rocket has to be 2 cubic feet, and it's a little bit smaller once it gets deployed at the International Space Station. As I've said, this is actually happening within a relatively short period of time, because we already have scheduled launches, first one being in November under SpaceX 16, and followed by another set of projects being launched in SpaceX 17 in February of next year. So partnerships with key stakeholders can rapidly accelerate your technology. Now, in terms of potential market for organs on chip, essentially the preclinical pipeline is about a $300 billion enterprise, expected to rise to almost $500 billion by 2020. All of that based on cell-based assays, laboratory animals, and a number of dedicated efforts for targeted therapy development. There's been a number of commercial activities around organs on chips, essentially within the last 5 years, 6 years or so. There's been a plethora of startups and other companies already trying to gain entry into this space because of certainly the commercial potential of this technology, not only in advancing drug development, but potentially replacing animals testing in the future. Where we're heading, I'll just give you in the last few minutes where we're going with organs on chips technology, certainly with advancement in IPS technology. What we would want to do in the future, relatively near future, would be to advance the technology to the point that we can actually do clinical trials on chips, which would actually inform the clinical trial design of interventions in people. And also, as I've mentioned, precision medicine, advancing the technology enough that you can actually put individual cells cells from individuals into these chips. And so in this regard, we were actually fortunate to capture some of the HEAL money, which is the helping to end addiction long-term, a big effort by NIH to address the opioid crisis. So under this guise, we were able to get some money to actually develop models for pain and opioid addiction, or at least plan to develop tissue chips for pain, nociception, addiction, and overdose. So that would be a funding opportunity announcement that'll be coming up within the next few months. I'll skip this because this is more about 3D bioprinting. So this is the effort to use tissue chips to model pain and opioid addiction. And as I mentioned, over the next few months, we'll be launching a new program to do clinical trials on chips, essentially to inform clinical trials. So this would be something that we are also keen on working with pharma, with FDA, and with patient groups, especially with rare disorders as well as with cancer and neurological disorders. With that, I'll end with this future in terms of applications of tissue chips. I already mentioned about clinical trials, personalized medicine, but certainly can be used with microbiome, looking at environmental toxin exposures, infectious diseases, development of countermeasure agents, and then advancing tools for clinical trials. So with that, I'll end with information, certainly a lot of information. All our investigators and the staff that supported this program can be found at the NCAT's website. You can also follow us on social media to learn more about this program. I think we have time for a few questions, so thank you. Anyone?
Dan, could you say a little more about clinical trials on chips? Does that mean that people would actually be enrolled in clinical trials and then you take fibroblasts from them and test your new compound on those chips?
Yeah, so that could take actually several different forms. So what could actually envision taking an ongoing clinical trial where there's an active compound being tested, and to take cells from those individuals and run the studies on chips in parallel and see if the chips can actually predict the human response, both in terms of safety issues as well as efficacy. The other application would actually be to start thinking about rare diseases where by the fact that they are rare, patients are hard to come by, and most of them are going to be very familial, and most of the time they're aggressive and pediatric cases where most of the patients have passed on. But if those patients have cell lines that have been banked, you can actually represent those cells on chips and essentially virtually expand the number of samples that can be tested or put on trials on chips. And so that would address the patient needs and then also preserve the naive status of patients, because as most rare disease patients are desperate for treatment, but they would almost always grab onto the first available one, but would compromise their status in terms of the next drug because they're no longer naive. So in this case, we could potentially conceive of looking at rare disorders, like I said, with no animal models, very few known about the disease, no natural history data. So I think there was mention last night about using AI as a placebo on a chip. So here's an opportunity. You can actually have cells from patients, a treated group and an untreated group, and be able to have a properly controlled study. So those are the kind of things we're going to be looking for. Yes, Christine.
Thank you for such an interesting talk. Over the course of a human being's life, their organs change, their cells change. So how do you cope with that? I mean, the organs of a child, let's say 0 to 18, are very different than the organs of a fully mature adult or an elderly adult. So
Right.
How do you cope with that?
That's actually an excellent point. So fortunately, we have sort of like a stem cell issue. It's been sort of like a conundrum for the stem cell biologists in terms of when you make fibroblasts into pluripotency and then differentiate them, they actually remain at early differentiation or juvenile state of differentiation. And then you have to coach them to become mature. So here's an opportunity actually for us to be able to look at developmental changes for drug metabolism. So taking the same cells and then differentiating them and letting them perform on the chip in the juvenile state with the epigenetic information and seeing how that changes when you mature them. So that's actually an information, because as we, as you probably know, that many of the drugs too are not tested in the pediatric population because of ethics issues. So here's an actually opportunity for us to look at those kind of signatures that would inform drug metabolism in the very young population. Yeah. Yes, sir.
A lot of disorders have a heavy environmental component. So do you have a program or thoughts about using your platform and the media, the environment there to model different environmental components on the disease, not only for cancer, where obviously radiation and so on, other things, but even in psychiatric disorders, things like bathing the cells in cortisol or something like that would sort of influence their reactivity. So do you have any thoughts on that, on modeling the environment?
Yeah, so we certainly have a strong interest from the EPA, NIEHS, Environmental Health Sciences, in terms of looking at environmental toxins, especially neurotoxins that would lead to developmental disorders. Also things that are estrogen-releasing things can be tested in the system. So we have actually partnerships with those agencies in terms of looking at those kind of issues in terms of exposures. The gut is also a very interesting study in the sense of looking at the microbiome. So we actually have been able to culture gut microbiome on the chips, and have been shown in terms of what those differences are in terms of its contribution to drug metabolism. And so that is still something that we can look more heavily, but has not been as mature as we would like, but certainly active area of research at this point. All right. I think I'm out of time, so thank you for your attention, and be glad to show you the chips.
That's fun. Thank you. Dan, thank you so much. What a great talk. So tissue on a chip, organs on a chip, human on a chip. I may not be the smartest tool in the shed, but don't think I don't see where this is headed. You can see how sneaky Lori and the BioCrossroads team, based on the way they surprised Anantha. I know what's going on. There's a vast conspiracy to work with Amazon to have some unit come up here next year to be your MC. So don't think I'm not following this along. Seriously, thank you so much for being here. Thanks for coming. Thanks for all the great work NIH does in so many areas, and the partnership is so critical with academia and industry. That public-private partnership in this type of discovery is just so important, so we really appreciate that. Thanks also for being here for Anantha. Anantha, congratulations. You know, you and Gus shared so many similarities, but the one thing that I think is most notable you both share is you're just both wonderful human beings, and you care so deeply about everyone around you. In all aspects. So couldn't be a more deserving winner of that award, so congratulations. So we've reached the lunch hour. I'm sure you're all excited about that. It's been a great morning. A couple final things here before lunch. Number one, I wanted to call your attention, you may have noticed this, but Indiana Oxygen Company right outside is giving away a Samsung Galaxy Tab A. And you just need to drop your business cards in the fishbowl out there, and they're going to do it immediately if you're interested, because they're going to draw the winner here during the lunch hour. I also wanted to mention, I meant to mention this before, the slides from most, if not all, of the presentations will be available electronically on the BioCrossroads website, right, Lori? Yep. So it's been a great morning. We're going to convene for lunch. So your lunch will be in Ballroom 4. Just out this way, I believe, and we're going to be back in here. We want to be back in here right at 12:30. We've got a great keynote capstone keynote presentation prepared for you. You're not going to want to miss that. And we also have, we'll close with some exciting information from BioCrossroads, an announcement that will be wonderful for all of us in the Indiana life sciences economy, if you will. So really exciting yet to go. So have a great lunch, and we'll see you in just a little bit.